So next up, we're going to move on to Elliot Lefkowitz, and you should be a presenter now. Elliot is at the University of Alabama in Birmingham, and he works with Ictv and as well as advises us on all things virology, and he will be talking to you today on the International Committee on Taxonomy of Viruses.
Okay, thanks, Anna. Can everyone see my screen and hear me?
Yes, perfect.
Thanks. Okay, so as Anna indicated, what I wanted to do is talk about the Ictv, the International Committee on Taxonomy of Viruses, and talk about its role in virology, how it does what it does, the resources that it provides, and then relate some of this to the current monkeypox outbreak and questions that have arisen from that outbreak. Okay, so taxonomy equals the process of both classification of living organisms or viruses into particular groups and then naming those groups. And so the resulting taxonomy should reflect the evolutionary history of the viruses that are being classified in the case of the Ictv. That's something that's been implicit in what the Ictv does, but in a paper that we'll have coming out within the next few months, we'll make that process explicit in terms of recapitulating the evolutionary history in the taxonomy of the viruses. So the Ictv is charged by the International Union of Microbiological Societies with developing and refining a universal virus taxonomy. And so the Ictv therefore provides the official taxonomy of viruses for use by researchers, public health people, governments, regulators, and also the general public. The Ictv has been doing what it does for over 55 years, first starting in 1966, and thereafter issuing a hard copy published report every few years up until 2011. Now everything we do is online, and so our online report is available from the Ictv website, ictv.global. So in order to do what it does, the Ictv uses a variety of methods of classifying viruses. Initially, of course, back in 1966, when these first discussions began, there were no complete sequences available in order to use for classification. So everything was done based on phenotypic properties. But over the years, genotypic properties, the sequence, and derived properties from the sequence have more and more been utilized in order to classify viruses. We recognize, 8 different taxonomic ranks now that viruses are classified, with the highest rank, the realm, being an implied ultimate origin of the different virus groups. So for each separate realm, the implication is that that defines a unique origin of the viruses that are classified into that particular realm. And then we have intervening ranks all the way down to the species, which represents the lowest level of classification. Importantly, the Ictv only goes down to the species level. We do not classify viruses at the subspecies level. When you see subspecies classification, that's done by the particular virologist working within a particular virus domain. There are also intervening subranks for each one of these ranks. So with regards to monkeypox virus, this is the classification of monkeypox from the species level, the species name being monkeypox, genus orthopoxvirus, and so on up, being part of the realm DNA viria. And so in order to determine what constitutes a unique species and on up, a unique genus, a unique family, the Ictv defines demarcation criteria. And these demarcation criteria are defined by individual study groups that are formed usually at the family level. So each family, each study group that works on a particular virus family defines the criteria that are used to classify the viruses in the particular groups. And so if we look at the demarcation criteria that come into play for pox viruses in general, at the realm level, DNA viria, the presence of a vertical jelly roll major capsid protein is a defining criteria for that particular group. Importantly, pox viruses actually don't have a icosahedral capsid, and therefore they don't have a vertical jelly roll major capsid protein that makes up the non-existent icosahedral capsid. But they do have a protein that appears to be an evolutionary descendant of the vertical jelly roll major capsid protein. But then on each of these additional ranks, you'll see different criteria that are used that are defined for the rank. So double jelly roll major capsid protein at the kingdom level. At the phylum level, these are all this group of nucleocytoplasmic large DNA viruses, including the megaviruses like the mimiviruses and such. And then there are groups that are mostly specific for poxviruses. The class at the Poxviridae, that also includes the Asifar viruses, African swine fever virus. And then poxviruses, the Poxviridae family, is unique to the order Cheetoviralis. Now, importantly, within the Poxviridae itself, both phenotypic and genotypic criteria are used for the classification. So host range is a major criteria that can be used to classify poxviruses. Growth characteristics, disease, serological characteristics can all be used as demarcation criteria at the different levels. At the level of the subfamily, to distinguish between the Entomopoxviridae and the Cortopoxviridae, the insect viruses and vertebrate viruses, host range, of course, is the primary means of classification. But actually, as it falls out, sorry, I'm getting a little pop-up here. Mostly these days, of course, as I indicated, genotypic criteria are used as the demarcation criteria. And these criteria can vary depending upon what level in the Poxviridae family you're looking at. If you're looking at the whole at the family as a whole, then amino acid sequence comparisons are frequently used, both in terms of looking at percent differences, but also in phylogenetic analysis. You have to use amino acids at the family level because of the AT sequence bias that differs between different genera in different in the different poxvirus species. But within the orthopoxviruses, nucleotide sequence identity within the core conserved region is a primary characteristics, with different species ranging from 96% to 98% identical, with isolates within a species greater than 98% identical. But most importantly, really, is phylogenetic analysis. And that's how a lot of the major decisions are made within the Poxviridae as to whether or not a particular virus belongs to an existing species or a new species, existing genus or new genus. And so we have defined here all of the different genera that are currently defined for the Poxviridae family, both in terms of the entomopoxviruses, the insect viruses, and the vertebrate viruses in the Cortopoxviridae. And so obviously we're concerned right now with monkeypox virus, which is a member of the genus Orthopoxvirus up here. We will most likely, there are new viruses that don't fall into an existing species or genus, and within the next year, the Ictv Poxviridae study group will be looking at the classification of those newly isolated viruses of the past several years. Here we have the orthopox species tree, defining all of the unique species within the orthopoxvirus genus. And you'll also see here that monkeypox with the two clades are present in this part of the tree, variola virus up here. Interestingly, you'll see that cowpox virus is present in multiple places in the tree. This is because viruses that we know to be, that are now called cowpox virus are actually polyphyletic. There's not a single lineage of all of these viruses that are named cowpox virus. So that really is a problem with naming as much as it is with taxonomy, and that's another issue that the Poxviridae study group will be looking at over the next year. So as I indicated, a number of questions arise as we look at monkeypox virus in relation to taxonomy. One is, do monkeypox viruses from the current outbreak, that is what we're calling clade 2b, represent a new species? And the answer to that is very definitely no. When you look at the branch lengths here between the clade 1 and clade 2, and then clade 2a and 2b in the monkeypox lineage, you can see here that those branch lengths are equivalent to, for example, the branch lengths that distinguish between variola-virus minor and variola-virus major. And so the branch lengths that represent distinct species going to the variola virus or camelpox or monkeypox viruses distinctly, the clade designations do not reach the criteria necessary to call them a different species. Another question that has arisen a lot is names and who's responsible for different names. And so quickly, names of diseases like monkeypox virus or COVID-19 are derived by the World Health Organization, and they're the ones in their ICD codes that define disease names. Virus names really come from the discoverer of the virus. And so whoever discovers it tends to name it, though there are certain exceptions to that. Clades really come from the scientists and the domain experts working on a particular virus. But as we've seen, the WHO has gotten involved with clade names for SARS-CoV-2, and then also more recently for monkeypox virus, defining the IIA, IIB, the Roman numeral clade designations for monkeypox virus. Finally, of course, the species names and any other taxon names are the responsibility of the ICTV. So when one talks about names, the ICTV is only responsible for taxon names, not for virus names or clade names or disease names. So as I indicated in the beginning, there are a number of resources that the ICTV provides from its webpage, ictv.global. You can go onto this page. You can create a membership that lets you make comments and such and get you on the ICTV distribution list for newsletters by clicking on the login link up here in the upper right. And then there are a variety of call outs for some of the different resources that are available. In terms of finding a particular taxon based on some name, you can use our taxonomy browser, typing in a name like monkeypox here, and then if that name happens to be part of an existing name or a past taxon name, if you check this little checkbox, you'll get an indication down here of what was found. And if you click on view, that will take you to our taxonomy browser, showing us here the context of the whole Poxviridae family. And then if we zoom in on the orthopox viruses, you can see the currently defined species within the orthopox virus genus, including monkeypox virus here. If you click on any of these rows here down by the little I, you'll get an indication of the complete history of the particular taxon that we're looking at. So in case for monkeypox virus, you get its whole lineage here. The last time that anything changed for monkeypox virus, the species, was in 2019. And you see here that the species was originally defined in 1971 as a part of what was then called the genus Poxvirus. In 1974, the family was defined, Poxviridae, and then the genus was renamed Orthopoxvirus. And so this history shows you an indication of all of those changes. In this case, the monkeypox virus itself, the name hasn't been changed. There was a part of renaming all viral species to a binomial nomenclature that starts with a genus name and ends with some unique name. We'll be renaming all poxvirus species within the next year. And then finally, I mentioned the Ictv report that used to be this big, thick, hard copy volume. Well, that report is now online. If you go to the link for the report, you see a page such as this that takes you to the individual report chapters, one for each family in general. Here's the report for herpes viridae, in which you have the table of contents. To the left, you have a nice description of the family and all its characteristics, and especially the characteristics that go into its taxonomic designations, and a number of figures that help with that information. We don't have a current online new chapter for the Poxviridae, but we will perhaps by the end of the year. But we do have the chapter from the 9th report, which was published actually in 2012, and that is available online. So let me stop there. Thank the ICTV and the executive committee for the work that everyone does, and all the subcommittee and study group chairs, and then also the group that helps me put together the website: Don Dempsey, Curtis Hendrickson, Alexei Shipinoff, and Donald Smith. And I'll stop there.